there were a lot of questions around host genetics and host genetics restricting responses to infections, including influenza as was a series of questions. And what I was going to do was show you some slides from one paper about host genetics and HIV that I think represents quite a lot of where host genetics and infectious disease work has been, and is one of the better papers. And then I have some slides on the flu question specifically. But so if people have questions about this and how to incorporate it into modeling, we can talk about that in detail. So where a lot of host genetics has really focused around infectious disease modeling has been around the role of the HLA. And I just wanted to remind people, since we've got a lot of new immunologists in the group, what the HLA is. So these are HLA is the human form of the MHC protein, which are the compartments for holding peptide antigens that are on every cell surface in the case of class 1, and on professional antigen presenting cells in the case of class 2. And so again, remember, we're looking down at the molecule here, and this is the cleft where they process and present peptides. And these proteins are the most polymorphic locus in the human genome. And so if you do any kind of genetic study, you almost always will pull out the MHC locus as a significant association with whatever it is you're looking at, because it is virtually unique to people, to an individual. And so you have, in humans, we've got 3 MHC class 1 molecules. I'll just scroll through these slides real briefly to get to class 1 versus class 2. And they're called HLA-A, HLA-B, and HLA-C. And then you have several class 2 molecules, some of which are classic antigen presentation molecules, and some of them are actually sort of more involved in stabilizing the class 2 complex and processing and presentation. Let me get my pointer. And the difficulty actually on the genetics of the class 2 side is that they are 2-chain molecules. They're both 2-chain molecules, but in class 2, both chains are potentially polymorphic. So you have an alpha chain and a beta chain for the class 2 molecule. For the class 1 molecule, you just have one main chain, and then you have the small molecule called beta-2M, which is universal for all class 1 molecules. And so it doesn't change between people, and it's actually quite conserved between humans and mice. You can actually use them interchangeably. And so for class 1, we're really only worried about the polymorphism of the A, B, and C molecules. For class 2, we have to worry a bit about the polymorphism of both the alpha and the beta chains for each of these molecules. And so for class 2, these are called DR, DP, DQ. Within the DR locus within class 2, there are multiple gene segments. And so you can have DR1, DR3, DR5, and in the same person. So you can have multiple DR molecules within one person. And there's quite complex linkage between, if you're DR1, you're more likely to be DR3 as well, and so forth. So I've mentioned the polymorphism. Many of you might be familiar with hearing HLA described in people, and so or typed in people. And so for class 1, you'll hear someone described as HLA-A, and then a 4 or 6-digit or 2-digit number. And so the most common HLA-A type is HLA-A 0201. There are thousands and thousands of different HLA molecules that have been typed across the class 1 locus and across the class 2 locus. And so a lot of cost in doing human studies is typing what HLA molecules people have, because that will determine then what peptides they can present. And so each of these HLA molecules has a slightly different motif of peptides they present. They're clustered in supertypes, so there's some homology among them, but you have to usually map epitopes for each molecule. Okay. And this is really critical because it changes not only what foreign peptides are presented, but it also changes what self peptides are presented. And so HLA molecules are also one of the #1 associations with autoimmune diseases. because as we learned on the first day, T cells are positively and negatively selected in the thymus based on their expression of self-MHC molecules, so they're self-HLA complexes. And so if you have an HLA molecule that's very efficient at presenting certain self molecules, and they're well expressed in the thymus, then you will have cells deleted that are specifically strong for those self molecules. But there's a lot of theory that there are certain HLA molecules and a lot of evidence in the literature that are not so great at presenting self molecules in the thymus, maybe because those molecules aren't presented in the thymus or they're not processed properly in the thymus. But those HLAs then go on to be associated with that autoimmune disease under the hypothesis that that peptide then is presented well in the periphery. And this is the concept behind this paper that I was going to show you in response to some of the questions on HIV. This is from 2010. It's from Arup Chakraborty, and it was in Nature. And Arup's a really good person to read for questions around T-cell evolution, T-cell selection. He's a quantitative guy, so he's a math modeler, but an immunologist. And I thought this was a really nice paper as it synthesized a lot of thinking and posed a real hypothesis that it then tried to test about peptide presentation, HLA, and associations with infection. And so one of the things that we know for HIV that's been studied really well is that having certain HLA alleles is protective or not protective for if you're HIV positive. And so in this graph down here, it plots 4 HLA types. They're all HLA-B types, so that's one of the class 1 molecules, A, B, and C. And it's plotting the odds ratio of whether or not you're likely to be a progressor or a controller, so a long-term non-progressor. And so there are these 2 HLA-B alleles that are associated with progression, so HLA-B07 and HLA-B35, and these are quite common alleles. And then there are these 2 HLA-B alleles that are associated with not progressing, with control and being a long-term non-progressor. And so this is HLA-B57 and HLA-B27. And again, there's 4 digits, there's actually additional digits on top of that. But we often just talk about the first 2 digits here. Okay, so what they were looking at in this paper, which was done with a statistical model of peptide presentation, was they took the motifs for each of these HLA molecules, because they're really well-studied HLA molecules, and so there's lists and lists and lists of peptides that bind to those HLA molecules, and they could infer a pattern of what makes a good peptide to bind to these molecules. And then they took all the peptides that they know that get presented in the thymus, and there's a fair bit of data on this, there could probably be more, and then they modeled how many peptides would be presented by each of these alleles in the thymus that would then be shaping the naive repertoire through positive and negative selection. And what they found was that these alleles that were associated with progression were more likely to present a lot of self-peptides in the thymus based on their model. So this is not empirically determined. This is a model of we've got this motif. We know what genes and proteins are expressed in the thymus, and so we're going to predict what peptides are expressed. And so the fraction of self-peptides that can bind to HLA molecules was higher in the progressors versus the non-progressors. And the hypothesis was that by presenting all of these different self-peptides and having this diverse set of targets for negative selection, the net result is that patients carrying these alleles are more likely to have a narrower peripheral T-cell repertoire, and that's going to translate into a reduced ability to mount diverse responses to different infections, potentially including HIV. And the hypothesis is that we can just really detect it in HIV because you have this very long time to develop a T-cell response, and the diversity of your HIV T-cell response, sort of like what we talked about with HCV, has been shown to be really important. And so there would be an advantage to having these HLA alleles that don't present self-peptides well in the thymus and allow a much broader peripheral repertoire to form. Does that make sense to everybody? It's sort of a very deductive argument, whether or not you follow all the steps. So there's a corollary to this. I wonder if anybody can guess. So among these 4 individuals, so we think there's these, among individuals with these 4 alleles, we think there's this association with protection from infection. So what would potentially the trade-off be? Just type it in the chat or shout it out. You've got an idea. Sorry, I'm looking for the Slack. I don't have it open. Let me do it over here. So self-protection versus HIV protection. So what do you mean by self-protection? I think you're on the right track. Autoimmune, exactly. That's exactly it. So the prediction then would be that if you aren't narrowing your repertoire in the thymus, you don't have a lot of negative selection, you're letting a lot of T cells out into the periphery, and one of those might end up cross-reacting with something that gets presented by self, and maybe you're more prone to autoimmunity. And the reason this paper probably got into Nature, in addition to Arup being a really good scientist and having a lot of good data that they used to make these models, is we already knew that these 2 alleles were really associated with the development of various autoimmune diseases like diabetes, type 1 diabetes, and so forth. And so it fit really nicely as a hypothesis that there's always these trade-offs. You can gain immune efficiency on one hand and protect yourself from infections, but it's going to come at the cost potentially of being more prone to autoimmunity. And this is this again, it's a 10-year-old paper, but this concept of balancing HLA and T cell priorities between self-protection and infection protection is really, I think, still a very productive area in immunology, and really understanding how T cells maintain tolerance and also break tolerance is really key. And I thought this was a useful thing to introduce because it relates to some of what I'm going to talk about in the influenza space. So I'm going to move on to talking about influenza. And I've put the references for everything on all the slides, and I can post the papers if that's helpful. I think they're all pretty much open access now because they're a little bit old. So the question was just generally what's known about host susceptibility to influenza infection. And this is a review from JI. This was an entire issue of reviews, or had a whole section of reviews about flu. And this is from Jakob Boone, who's at WashU. And this is a really useful paper, I think, because he went through PubMed and very carefully curated mouse data on every knockout mouse that had been tested for flu, and whether the knockout had a protective effect on survival, a negative effect on survival, conflicting effects in the literature, or no effect on survival. And what was interesting is this very long list of genes that had a protective effect on survival. So you lose a gene and you gain resistance to influenza infection. And so there's a lot of discussion in there about why this is so, and potentially interpretation around this. But this is not, I think, the kind of genetics that you all were asking about in terms of human host genetics and host genetic variation, but it's a good place to start because it gives you kind of a good reference for the genes and the pathways that, if you completely ablate them, have a particular effect on outcome after flu infection. And he tries to relate it here to how these different pathways might play in. And so, pathways could affect pre-existing immunity, which will control viral titer. If you have no immunity, then you have this sort of unrestrained viral titer, as we talked about yesterday. And then there's all of these other host factor features that can prolong inflammation and prevent wound repair, which is really the key feature of susceptibility to flu. Is the initial phase of infection can usually be controlled relatively efficiently, but the restoration of lung function and the dampening of immunopathology is associated with a lot of the severe disease. And genetic pathways likely play a role in this. So, this sort of summarizes this kind of artificial condition where we're knocking something out. But if you now look at human data and ask whether or not there's, evidence for a pathway to be involved in the response to flu, you can kind of reference back to some of the key mouse experiments and say, well, at least when that pathway is completely ablated, it has a positive or negative effect. I see the question in the chat, and I'll come back to it in a couple slides because I think it'll tie into a little bit of where I'm going with the flu stuff. Okay, so then moving into humans with flu and asking about human genetic associations with influenza infection. There's a much smaller literature here, and there's a much smaller literature really across humans and infectious diseases and genetics in general, even though it's still a reasonably sized literature. And the big name in this field is a guy at Rockefeller University named Jean-Laurent Casanova. And he has influenced the field quite a bit and has done a lot of very good work. And he's behind a lot of the studies I talked about yesterday with respect to TB and identifying the interferon gamma pathway as being absolutely critical to controlling mycobacterial infection. But he has a very specific point of view and a sort of theory of the world of host susceptibility to infectious diseases and how genetics plays a role in that. And that is that really severe, unusual susceptibility to infectious disease is primarily the result of monogenic catastrophic variation or Mendelian catastrophic variation. And so what I mean by that is an entire gene or entire pathway that's been ablated in some fundamental way, and probably occurring at extraordinarily rare frequencies in the population. And so that is mentioned here. The monogenic infection is rare here. And he talks about Mendelian infections here as being common, but this is common in sort of a very narrow sense of what common means. It's still quite rare by our general standards. And so the idea is that if you have an otherwise healthy individual that, say, dies of the flu or gets very sick from the flu, and similarly, COVID, he expects that he should be able to find a genetic lesion in that individual that's knocking out an entire pathway, basically creating a knockout mouse type phenotype, and that it'll be sort of extraordinarily rare and a one-off in a particular person. And backing this up, he's published a couple of papers on flu. So one of them was on IR7 and another on TLR3. And in the IR7 paper, it was a young woman who had a very severe flu infection. She was otherwise healthy. She had never had any other bad infectious episodes of any virus. I think she was somewhere between 5 and 10 when this flu episode occurred. And they sequenced her genome, and she had two different deletion mutations in her IR7 gene. And this is one of those key transcription factors that mediates the type 1 interferon response, which we've discussed over and over again as being really critical for viral infection responses. And so one was inherited from mom, one was from dad, and they were totally different mutations. And they were extraordinarily rare mutations, so like 0.001%, each of them prevalent in the population. And so the odds of that genetic cluster ever occurring again in the world is, infinitesimally small. It was just, sort of a bad luck scenario. And this is his view of how genetics influences infectious disease, and that'll make it incredibly difficult to predict because it's extraordinarily rare, and then it's also targeted to, he thinks, very specific infections. And again, this is a very singular view, but it's a very prominent view just because he's a prominent person in the field. And so for COVID, for instance, he is leading an international effort that children that get extremely sick from COVID, and even young adults up to 40 or so that don't have any underlying conditions that get very sick with COVID. He wants to sequence their genomes, and he's going to report that that they have these catastrophic mutations in these different pathways. And I'll be very open about my point of view. Is it's a difficult hypothesis to confirm because he never publishes the ones where he doesn't find the mutation. So you only get these papers where he finds a mutation and it's very clear, like IRF7, that just screams out at you. Of course, this is going to be problematic if you get a viral infection. And, he might have sequenced 100 other individuals and not found the causative deletion, and he doesn't report it. But it's led to this point of view that it's these rare variants, because what these are called then are rare variants that are really driving population level susceptibility to severe disease when it's a rare event. There's another view, and they're not all mutually exclusive, but he's a very dogmatic kind of voice, and so sometimes it can seem like they're mutually exclusive. And if anybody is in the Casanova camp, I'm not trying to be overly critical. I'm just sort of presenting my point of view. But this is a review from a sort of independent group looking at genetics of flu, and you see IRF7 there. That's derived from the Casanova work. And you see some other genes as well. And one of the other genes that's been heavily studied in flu, and in full disclosure, my lab has contributed to this literature as well, is IFIDM3, which is one of those interferon-induced proteins that restricts flu virus replication. And the variants that have been associated with IFIDM3 and its expression and susceptibility to flu are actually very common variants. And so they're widespread in different populations. They're associated with different ancestral populations. So some of the variants are very common in Southeast Asia, and some of the variants are very common in Europe and in Central and South America. But they are not the rare variant sort of point of view of Casanova. But they also don't have the effect size of those rare variants that Casanova finds. And so that's the trade-off, is that when you've got a catastrophic rare variant that just knocks out a pathway, then the penetrance is potentially going to be really high, or the effect size is potentially going to be really big, because you just cannot make IRF7, or you just can't make a particular cytokine, interferon gamma. The common variants are usually things like in a promoter that changes the expression level of a protein. So you have twice as much as you had before, or you have half as much as you had before. And it makes you slightly more susceptible. And so it, in combination potentially with other common variants, will lead to a susceptibility profile that puts you on the far end of the spectrum, and you're more likely to struggle with disease. This is very hard to tease apart, though, because there's so much noise in these human studies, and finding the individual contributions of each of these common variants is still actually somewhat controversial statistically and mathematically in terms of how you do it, and even experimentally in terms of how you validate it. And one of the things the field has sort of coalesced around of what needs to be done to sort of have confidence in these common variant calls is that we have to figure out the mechanism that the common variant works by. And so this has come out of sort of one arm of the reproducibility crisis in science, is that there were a lot of these large genome-wide association studies where they would just report a single SNP as associated with a particular disease. It would have a very good p-value. But then when they would go to repeat it in a different population, or in a slightly different disease group, or in a slightly different disease severity call, it wouldn't repeat. And so the idea is that if you're going to identify a common variant and find a SNP, if you can then say the reason this common variant SNP is important is because it is in the promoter, and it changes the binding of this transcription factor. And so this transcription factor doesn't bind, or it binds now a different transcription factor. And we show that it's what's called an EQTL, so an expression variant that changes the expression of that gene. Then we can have a little bit more confidence that maybe we're finding something that looks like a mechanistic insight into genetic susceptibility to disease. It's still not foolproof. You could still be wrong. You could be right about the common variant changing the expression of the gene, but you could be wrong about that change in the expression of the gene contributing to the severity of the disease. And so just replication across cohorts and building cohort studies to really consider and take consideration of these variants is really critical to pan that out. But if you read this literature, you'll see this tension, and then you'll and it makes it a little bit harder, I think, for modelers to try to think about how to take account of these things. This is from that same critical care article that I just on the last slide, and it curates as of last fall, I think, all of the pretty well curated human common variant SNPs for influenza. And so IFIDM3 is there at the top. CD55 has been shown in a number of studies. TMPRSS2 is one of those enzymes that can cleave the HA, and that is restricted to the respiratory tract. Interestingly, TMPRSS2 is also involved in the life cycle of SARS-CoV-2. And you can go down the list, and you can see while not all of these have full-on mechanistic confirmation of how the SNP relates to a particular outcome, they're all in pathways that make some kind of logical sense in terms of their immune pathways, their lung pathways. They're not sort of a kidney gene or something like that that would be difficult to associate with flu. And then on the last slide, I just want to point you to this review, which is really nice. This is from 2012 in Nature Reviews Genetics. And it kind of gets right into the heart of this argument between the sort of rare variant camp and the common variant camp. And it presents the top 20 arguments saying, for and against rare alleles as being the main drivers of disease, and for and against common alleles as being the main drivers of disease. And then it presents this sort of fusion view, which makes sense of the potential joint effects of rare and common variants. And this is in metabolic disorders. And it's suggesting that for rare variants to really have the penetrance that they have in certain individuals, they have to have a collection of common variants that sort of open that pathway up to the susceptibility of catastrophic damage from a single gene being a variant in a particular way. And if you think about that, that just complicates things even further, because it means that we have to sort of try to keep track of rare variants and common variants. Rare variants are obviously intrinsically hard to study because they're rare. And so finding enough people that have any individual rare variant is probably going to be impossible. As I told you, that the individual with the double IRF7 mutation is probably never going to occur again in human history. And so that's a one-off. And we just have to accept that it's so biologically plausible that that's probably the causative or proximally causative to her susceptibility to flu. But if she has an additional set of common variants that are affecting that susceptibility, it's going to be impossible to tease that out because we'll never have replication or a control group. So that's what I was going to tell you about in terms of flu and genetics, and just genetics and infectious diseases. It's I think a really growing area because we have so much more genetic data, and it's so cheap now to get genomes, and we really are trying to understand this. COVID makes the case better than anything. If we could figure out prospectively among healthy young people who's particularly susceptible to COVID, that would really change at a public health level what we might do in terms of, say, opening schools or putting people in groups where they can work together. If you knew with certainty that the worst outcome you were going to have from COVID was, a mild cold, that would probably change a lot of our behavior. And while we can identify, the big demographic groups that have susceptibility, everyone's worried about that, reasonable percentage of young, healthy people, stories you see on Twitter of people that are highly susceptible. And I think we're going to see this rare and common variant argument really play out intensely in the context of COVID as we try to figure out what really underlie that susceptibility. Okay, so then getting to this question, do we see or expect the population frequency of these two types of HLA haplotypes differ between human populations that are under a different HIV prevalence? So I think what the question is are we starting to see selection on the human population by HIV? And the answer, I would say, would be, or is HIV prevalence in particular populations driven by the carriage frequency of some of those HLA haplotypes? And not to my knowledge are there any clear signals that an entire population is you know, sort of been spared because of their high carriage frequency of a particular HLA molecule. Now there are population level studies of HLA carriage frequency in influenza and susceptibility, and I can provide you references to those that suggest that, you know, certain HLAs are not great for dealing with influenza peptides, and they're at high carriage frequency levels in populations. Again, it's difficult to tease apart all of these, all of the potential noise in those studies. The HLA allele that has been repeatedly shown to be a susceptibility allele for influenza is A2402, and A2402 is commonly found to be a susceptibility allele in a lot of viral infectious diseases. And A2402's one of its distinguishing features is that it's a generally high carriage frequency in indigenous populations around the world. Interestingly, it's sort of almost any indigenous population. So indigenous Australians, Pacific Islanders, the Maori in New Zealand, but then the indigenous populations in Alaska and the US, the indigenous Native American populations in the United States, and then the indigenous Central and South American populations all have A2402 high carriage frequency, and they are all susceptible to influenza, actually at higher rates. But obviously there are a lot of socioeconomic and other health factors that characterize those populations, potentially different factors, but still that may put them at risk. And so this is the problem with genetic studies and HLA studies in particular, because they can identify these populations and do identify these populations. And so if an entire population has a number of other correlated risk factors, teasing apart those correlations can be a little bit difficult.